Thank you, Elliot. Today I'd like to share with you how Decibel is using cloud-based infrastructure to drive development of therapeutics for both hearing loss and balance disorders. So, quick background on Decibel. We're a biotherapeutic company focused on hearing disorders and balance disorders. This is pretty much an open greenfield area right now. We have the first integrated drug discovery, development, and translational platform that's focused on the inner ear, and our approach is to be modality agnostic, so biotherapeutics, small molecules, gene therapy, whatever works best. We are about 4 years old, located just down the street on Boylston, next to Fenway, about 80 FTEs and growing, and we're still a private company. All right, great. So here's a few high-level statistics on both hearing and balance disorders. 1 in 10 individuals suffer some form of hearing loss in their life. That equates to globally 500 million people who are impacted by this. Children oftentimes born deaf at birth. They have delayed cognitive, emotional, and social development. Age-related hearing loss is also a risk factor in the elderly for dementia and other cognitive declines. Altogether, it leads to lost productivity, anxiety, depression, and a host of other related issues. On the flip side, this sort of blew me away. Every 20 minutes, an older adult dies from a fall. So think about that, and then just think about all the falls that don't result in death, but also result in serious injury and what the impact there is. So overall, worldwide, this is a tremendously unmet need, and if we can do anything to address this, we can have a significant impact on both quality of life and even survival. Oh, there we go. So how do we think about Decibel in terms of how we organize to address this unmet medical need? We think of 3 primary disciplines that are interdependent. First, hearing biology. So on the flip side, maybe one of the disadvantages of being in a greenfield area is you can't build a team by just drawing from other groups at established biopharma, right? So we've had to grow our hearing biology group and our capability organically over time with internal development. Same is true in the clinical area, right? Conducting clinical trials on hearing indications, balance indications. This is new territory. So as a company, we're learning how to do this, and as we enter into the early phase 1 trials that we currently have undergoing. Drug R&D, like I said, modality agnostic, small molecules, gene therapy, et cetera. One of the interesting things about the ear is it is contained somewhat similar to the eye. So one of the first approved gene therapies was in the eye. So there are advantages there in terms of containment and also accessibility. So we're working very carefully on formulations and delivery mechanisms that can take advantage of that fact. And that's true, for example, for one of the compounds in our clinical trial, which is administered locally. Finally, data sciences. To support all of this, we have a strong computational genomics group that's been there for couple of years now, and now this past year we're investing in software engineering, informatics, machine learning, et cetera, to build up that capability and take the data that we've already generated, aggregate it, integrate it, and see what additional insights we can gain from that data. So this gives a snapshot of the capabilities that we're using in our new product engine. There's a strong focus on human genetics using data sets like the UK Biobank, mouse genetics with forward genetic screens, also using transgenic models, mice, CRISPR knockouts. We're doing a lot of this in collaboration with Regeneron. That's a strategic collaboration. It affords us the opportunity to leverage capabilities that we wouldn't normally have had as a small biotech. And that collaboration is working very well. Single-cell RNA, we've sort of been a leader in that area in terms of utilizing that in hearing. We have the largest library of gene expression for inner ear, spanning various in vivo mouse models, treatment control conditions, mouse strains and knockouts. In situ transcriptomics, basically the ability to look at what's happening at the cell morphological layer globally, understanding where certain genes are expressed and what the impact is on cell death or other issues. As I mentioned before, in the drug development and optimization area, we are modality agnostic, but then there's advantages with route of administration where we can locally administer either small molecules or gene therapies. So we're always looking to let the science drive the platform development. In this sense, it's broadly the platform development. That includes our platform for gene therapy, where we've been able to utilize our single-cell RNA expression data to both identify and select unique promoters for particular cell types that are relevant for the genes we want to target in particular substructures of the ear, how we deliver those therapies to that inner ear, and then using big data and cutting-edge informatics on large-scale data sets like the single-cell RNA and the genetics data sets to identify new targets. So I'm going to walk through sort of 3 data domains that we think about that are critical for us. And the first one is a molecular profiling repository. So DNA variation from genetic data sets, single-cell RNA expression, ATAC-seq to understand how accessible the genome is in particular cells, which can guide in terms of how you want to use promoters and enhancers. And then cell morphology, we do a lot of in-house imaging on all of our in vivo animal and mouse models. In that case, that's a real driver for what we need to do from an infrastructure perspective because we need to get all of that data back up onto Amazon and then store it and analyze it there. The theme around the molecular profiling is that all this data is at scale. So it's rather uniform in some respects, but then you have to deal with the fact that each of these data sets can be terabytes in scale. Terabytes for genetics, terabytes for images in aggregate, and terabytes for our RNA expression data. On the clinical side, we're conducting our own clinical trials, and those include both interventional studies and observational studies. Full disclosure, I'm actually participating in one of our observational studies around tinnitus, basically when you hear, say, for example, buzzing in your ears when there is no sound. So they're trying to figure out, are there correlations with people who experience tinnitus and other sort of life factors, et cetera. And that was sort of eye-opening for me to be on sort of the other side of things and actually go through a full audiometric workup and be a participant in an actual clinical trial. In terms of data that we're getting externally, we're partnering with Regeneron and Geisinger to get data sets, clinical data sets, Karolinska Institute in Sweden, Mass Eye and Ear. All of these data sets we're then cataloging in our clinical phenotype repository. So the challenge here really isn't the scale of the data, it's the variety of the data, right? So each of these data sets is slightly different in terms of its composition. And these just highlight the largest data sets. We're also going after smaller sets of data of moderate size from different academic collaborations. So the first goal is to catalog it all. The second goal is to prioritize and decide, are there ways that we can conform the data across all these different data sets on a prioritized basis to understand if we can ask and answer questions across these data sets. So hearing loss has some unique data streams. One of them is all of the audiometric data that you need to capture when you're doing a clinical trial. So the gold standard right now is to use a sound booth to attenuate ambient noise and then use an audiometer with different mechanisms to test for hearing. That can be pure tone threshold test where you raise your hand when you hear the tone to figure out what the threshold is. You can also do bone conduction where that vibration in your bone actually travels to your inner ear and you hear it as a sound. That bypasses the outer ear and the middle ear, so it helps you understand where the issue really is, if there is one. And then finally, there's a mechanism where you can actually play two tones, for example, that are fairly close together into your ear, and then there's a little microphone, and that dynamic response of your inner ear actually emits, otoacoustic emission, emits a frequency that is some multiple the difference of those two frequencies. And so that's actually reveals something about the health of the inner ear that you don't get from the other tests. And so oftentimes, for example, if we're dealing with mouse models, right? Mouse can't raise their hand, I've heard the tone or not, so you need other ways to understand whether the hearing is actually working. So the challenge with the sound booth, two of them, one is that they're expensive, they take up a lot of space, and so you have limited reach in terms of the number of patients that you can reach. The second is the audiometric device market is highly fragmented, so that's a challenge there because there's different devices, each with their own proprietary software. So what we're doing there is trying to find ways to use potentially and hopefully commercially available systems that can act as a centralized and consistent way to pull data from each of those audiometric devices so that we can then directly collect that clinical audiometric data in digital form rather than, say, having a clinician print out something from the audiometer and then enter it into a clinical case report form. In order to address the issues with the sound booth, we're partnering with companies and looking at different companies that can provide headsets. There, the key is how well can that headset attenuate that ambient noise? And then also, what kind of tests can you do? Can you do bone temporal conduction tests, et cetera? So that will allow us to at least do assessments on a much broader set of patients. And then the hope long term is if you can get those headsets to do just as well as you can with a sound booth, you can ultimately replace the sound booth. And then finally, we're building a mobile app. So that allows you, and as an individual, to do a quick hearing test on, say, for example, your iPhone. That would be valuable for wide-based screening. Also, follow-up monitoring after visiting, say, a sound booth or a clinic. You can then, over time, get some consistent results about whether your hearing is degrading or improving over time. And it also can be a device just to generate educate the general public in terms of sort of hearing tests and maybe what things you should watch out for in terms of things that might impact your hearing. So our audiometry repository, we've named Earbase. That exists now. We're doing another rev of that as we speak. So that sort of wraps up the sort of the science and the data stream collection. Now I'm going to turn to the technology platform. So we're looking for flexibility. You can see all the different types of data sets and technologies we use. We're rapidly acquiring new data sources. Some of those oftentimes are large data sources within a matter of days. We want to get it downloaded, want to get it analyzed. And so, AWS provides that level of flexibility. Leverage, we're a lean organization. We're not that big. So we want a platform where we can do a lot with minimal overhead and minimal management, where it's easy to spin up capabilities, not just instances, but for example, clusters and other higher level order services. Usage-based, so we're focusing on serverless where possible. Classic example of that would be Athena, right, for serverless query acceleration of data that's stored, for example, on Parquet files on S3 or JSON files on S3. Using on-demand still works well for us, where we can spin up a large instance, do some analysis, put it in stop mode for a while. Geneticist wants to go back, do some additional analysis, just turn it back on and keep working. Fine-grained access for collaborators. So goal here is to bring the collaborators to our compute platform and to our data. So one of the issues is that with some of the sensitive data we use, we don't want to actually have to worry about that data streaming out. So we want to bring the collaborators into our environment for compliancy purposes. So Decibel, being a Third Rock Ventures funded company, started with the Biotech blueprint. The Biotech blueprint was spearheaded by Third Rock Ventures platform team. It provides a mechanism to spin up a complete AWS environment with VPN access and third-party applications, for example, Chemaxon, in a matter of hours. All the networking is set up, and so it's a huge win for small biotech, so you don't have to hire a team to go ahead and get that done. We're starting to outgrow our sort of simple blueprint right now. So we're looking at going to a multi-account model, so that will help support our software engineering efforts, where we have dev and test environments that we want to isolate, where we want to separate, for example, analytics and bioinformatics from an account of shared services, which might be our VPN connection, domain controllers, production assets like that. Multi-region. So with Amazon being available in places like Asia, what we can do is we can have our CROs, if they have larger data sets that are not just Excel files, but rather large images, we can have them upload those into another region where the network connectivity is much better, and then we can use the AWS backbone to actually replicate that data back to our US East region, where we could then analyze it. Direct Connect, we've outgrown our VPN over our 500 megabit internet connection. We'll be putting in place a Direct Connect that'll route through Markley, that'll provide some feature flexibility. We'll start with 5 gigs. And the key driver there is the in-house imaging, right? So the number of images that we generate on a daily basis, we need to be able to stream those up to Amazon much more quickly. And then there'll be cases too where we want to bring that data back down. In terms of getting the data up there, right now we're exploring File Gateway. We're using that on-prem. We're also using it in EC2. So there's a common theme here I've experienced both the work I've did before and the work here, which is this object versus file system. To me, it still seems to be somewhat of a pain point. You'd love to just work with S3 objects in general, but a lot of the tooling, genomics, image analysis programs, image management programs, they all want to see a file system-based backing store. So in that case, we need to put something in front of that like a file gateway. So this is our 10,000-foot view of the data science and engineering platform. The key here is it's all driven, for the most part, off of AWS. The number of components that exist outside of AWS is rather small, and that's by design. That's how we started with a blueprint. We'll have core services around identity management, DNS, et cetera, that we utilize across our total platform, for example, Office, Ignite, et cetera. We'll utilize those services in addition to Amazon. And then on-prem, we'll have the Direct Connect and the storage gateway, specifically the file gateway, to cache data from AWS objects, both directions, for example, right? You put something on that file gateway, it gets streamed up to S3. You can actually put things on S3. It'll get cached back down if you ask for it onto the file gateway. So right now, that's our strategy for providing that flexible interface between S3 and the file system on-prem. Starting from the bottom, S3, from a sort of a data lake perspective, is our primary storage, where we hope to keep all of our raw data, all of our refined data that comes out of analysis. It's low cost, scalable. It's integrated with other services in Amazon like Athena, Redshift Spectrum, Glue. You can use the model of database tables as objects with Parquet and Avro and CSV. And we're going to be taking a look in the next few weeks at lake formation, working with the AWS team. Just a quick recap, we use EBS, and we actually use EFS extensively. So EFS is sort of shared home directories, shared analysis directories, for example, for our notebooks and Jupyter notebooks, so that the scientists and data and informaticians that are doing the analysis can easily share data with each other, and it's a persistent file system. We just also took advantage recently of the fact that we can now back that up easily with Amazon Backup. We also use selected EBS volumes to cache data, for example, the UK Biobank data. If we need to have that on a file system, we'll quickly pull that down, put it on an instance, and then do the analysis with certain genetics tools. Our hypothesis is that we can go a long way with Athena before we have to go to something like Redshift or additional database, NoSQL databases like DocumentDB, DynamoDB, probably standardize on Aurora, RDS for our Postgres databases. And then we'll start to pull this together with using Glue as a technical data catalog with the Hive Metastore, where we ingest and catalog all the Parquet, CSV, and actually directly database connections. and then use lake formation to help with the management of that around integrated access control. And then finally, we have our Jupyter and our notebooks. We have our EMR. We have Hail up and running. We expect to sort of productionize what exists now as sort of genomic pipelines that have been developed by the CompBio team. We'll take those and productionize those and probably use a framework like Nextflow or SnakeMake for that. So even though we're a local company, all located here in Boston, we have global reach. We have clinical trials ongoing now in Australia. We have CRO partners, WuXi Chem partners in China, collaborators like Regeneron here in the US. We do all of our sequencing through the Broad, pulling data in from the UK Biobank, partnering with the Karolinska Institute in Sweden. So as I mentioned before, there's a lot of advantages to having AWS available in all of these regions to help facilitate data ingestion. Right now, we're using AWS to ingest all of our large-scale data, genetics data like UK Biobank, our imaging data, whole genome sequencing data, which we're doing with select collaborators on some of the clinical trials, and then using cross-region replication where necessary. We still make extensive use of Ignite, so that has a certain usability feature for collaborators where they can easily deposit files like Excel files and translational data sets that are more moderate size. One of the nice features of Ignite is you can actually just send out a link that allows someone to essentially drop files in a particular location on your Ignite file system, and they don't need access to anything else. They can't see anything else. All they can do is use that as a drop zone. And so we've used that both in the research area and the clinical area. The other thing is we're doing a pilot with Tetra Science where we take some of that semi-structured Excel data, we actually turn that into structured JSON, and then we can now load that into the data lake, import that into Glue and Athena. So a sketch of a data lake, it's simple for purpose of the fact that it's actually a work in progress. This is somewhat of a forward view, and to emphasize that we see sort of three key layers to our approach to managing a data lake. One is the basic storage, S3 where possible, file systems where necessary. Second layer is the query accelerators like Athena and other databases where, for example, we might restructure the data to support certain application access patterns where we want greater performance. And then finally, using Glue to pull all that together. Now at the top, we have three sort of applications, if you want to think about that. One is an actual application study tracker that contains the scientific metadata. So that's all about the experiments, the design of the experiment, what compound you used, what gene is relevant, what strain of mouse in an in vivo model. So that pulls together all of that scientific information and can have links out to, for example, PowerPoint reports, et cetera. On the right, data catalog contains the operational metadata. So that's a combination of the Hive Metastore, the technical metadata that you'll get with using Glue, and what we're going to augment around sort of who the data owner is for that data. Where did that data come from? Are there certain compliance requirements around that data? So we'll capture all of that in our data catalog. And then finally, building out a results management interface or database with analytic metadata. So for example, we have expression data, but then you can do analysis so you can get differential expression, you can get phenotype, genotype associations, PK results. We want to model all of that in terms of semantics of the different data types so that then we can know and understand how best to pull those together in terms of visualizations and additional query support for our scientists. Stuff on the right is basically how you take that raw data and then you turn it into those refined results. It's a combination of potentially notebook type use, dashboard use by scientists, plus clustering, batch, et cetera, all of the sort of programmatic work that we're going to do to systematize how we handle that data once we've worked with our bioinformaticians and our genomics and computational biologists on exactly what the right approach is. All right. So now I'm going to walk through 4 examples of sort of the type of analysis that we're doing on the platform now. One is a look at the single-cell map of transcriptional profiling within a mouse model. So this is one of the advantages of have that large expression data set that we've been collecting over the past couple of years. This is an example of looking at 80,000 different cells and being able to pick up, in some sense, smaller signals or much rarer cell types that are representative of what's in that fine-grained structure of the inner ear. So we have, the standard well-known cell types that everyone expected, but, we have more than that in terms of less-known cell types, things that you might not have expected or anticipated would be relevant for particular biology of hearing within a mouse ear. You take that, look at it in a slightly different way, and then you've clustered by genes across the different cell types. What this highlights is in the bright bars, you can essentially view those as a set of marker genes for the particular cell type. So we run our single-cell RNA expression data through our pipeline. We can then automatically tag which cell type that particular cell is based on the marker gene set. And then also based on the uniqueness of those marker genes within each of those cell types, we can use that, as I mentioned before, to inform how we can use cell type-specific promoters in, for example, our gene therapy constructs. So we took a look at the UK Biobank data recently, and we did this actually just using a large memory optimized instance. We had Hail available, but we didn't need it. And for this particular use case, the geneticist had tools that he was more familiar with, more comfortable with, and felt he could tweak to get the kind of results he wanted to. So he went ahead and used that and identified 22 new loci that weren't previously associated with hearing in the literature. And then even what's interesting is our 6 loci with known monogenic deafness implications that are also associated with age-related hearing loss. So if you have maybe 2 copies of the mutation, deafness, maybe 1 copy, you're more susceptible to age-related hearing loss as you grow older. We're doing a pilot with Aaron Harvey, who's actually giving a talk later today on leveraging NLP to enrich clinical phenotype repository or electronic health records. So one of the first things is got to remove the PHI. So we've taken a look at that, found some high levels of false positive rates. So examples are where doctors' names are identified as PHI, probably not. There's rules you can do to get around that in terms of just looking for the doctor prefix. Also name-like conditions named after individuals. I figure it'd be easy to back those out then when you do the follow-on thing where you actually pull out the conditions using Amazon Comprehend and then identify those. And so we're sort of trying to assess what's the impact, how much effort do we invest in rules, because if the downstream analysis doesn't really depend on it, then maybe you don't want to go through that effort. And then after that, we'll take and look at what we can do in terms of understanding correlates and predictors of hearing loss, identify at-risk individuals, and do patient stratifications. So here's an example of doing that, not on the electronic health records, but using a public data set, NHANES, the National Health and Nutritional Examination Survey. It's a much more highly structured database, so you can start with that. And then you can identify a known set of risk factors for individuals who are more susceptible to hearing loss. So you can see that as a function of age and the threshold. Lower thresholds are better, that high-risk individuals have much greater likelihood of a higher threshold or greater hearing loss. This is not a deep learning algorithm, but we didn't need to go there for that. So not everything needs to be solved with deep learning. Quick shout out to the data science team. So Adam Palermo leads our computational biology and genomics group. Dave Toomey, right over there, leads our software engineering group, and I head up informatics. And it's all led by John Kilty, who's our executive vice president of data sciences. And with that, I'm done, and thank you.